Well, good evening, everyone. Welcome to tonight's episode of the Late Night Conference, second season, episode number 5. My name is Wilhelm Huck, and I'm your host tonight, although, well, I'm kind of your host tonight, but I'm not physically present today in the Radboud Lecture Theatre, which is a bit of a disappointment, but I'm sure our speaker today will make that disappointment go away very quickly. As you know, this season, we have been talking about artificial life. And last time we talked about robots and almost sort of ways to make robots think like us, or at least make robots, well, make us believe that the robots were thinking like us. Not quite. But today we're going back to the sort of molecular level of building artificial cells, building synthetic life. And as you might know, in the Netherlands, we have a large consortium called Building a Synthetic Cell, BASIC. Well, as you know, the US is a much bigger country. And so, of course, they have a much bigger consortium, which is called Build a Cell. And one of the leaders of that consortium is our speaker here tonight, Professor Kate Adamala, who is a professor at the University of Minnesota. And of course, being in Minnesota means that she probably likes the cold very much. And that is probably true because she hails originally from Poland. She studied chemistry at the University of Warsaw. And then, well, I think she did an international PhD, working both in Rome with Pierluigi Luisi and with Jack Szostak at Harvard University, where she studied, well, I think origins of life, the RNA hypothesis, but also peptide catalysis and liposome dynamics, and published some wonderful papers on, well, very simple chemical systems capable of Darwinian evolution. After a postdoc in Ed Boyden's lab on synthetic neurobiology, I don't really know what synthetic neurobiology is, but maybe Kate will tell us a little bit about it. She joined the University of Minnesota, where she has rapidly built a very thriving research group. And so tonight, I hope she will be talking about her endeavors to build a synthetic cell from the scratch. I have to admit, we know each other very well, and we have been working together in the past. We just published a paper with 3 years of results that didn't work, but it was a major piece of work. And that's why it's my great pleasure to introduce Professor Adamala to you today. After the lecture, of course, on during the lecture, please ask your questions, put them in the chat, or store them up and ask them to Amy, and then we'll collect them all at the end, and we'll have lots of discussions at the end. But for now, Kate, the Zoom is yours.
Thank you very much. Thank you so much for having me. I just want to correct one thing for the record. I do not like cold. I hate cold. I just happen to end up in places that are cold. But I love the people and I love the work, so it's a privilege to talk to you. It's kind of intimidating to talk to you because Wilhelm and your research community, you guys are leading the efforts in engineering synthetic cells. So talking to you is kind of like me talking about baseball at the Super Bowl. And so I'll try to do a good job talking about it, but I'm really looking forward to the discussion. So this is actually one good thing about the pandemic is that I can talk to you without spending 8 hours on the plane, so I'll take that. So what I want to talk about today is engineering synthetic cells and why are we doing it and how are we doing it. So I try to explain kind of the rationale, the background of our field. So I want to start with answering the very obvious question. Why do we bother making artificial cells if we have perfectly good normal cells? In fact, you might argue that we have more than perfectly good normal cells. We have this amazing diversity of cells. We have a lot of different forms and functions seen in nature. There is a lot of different ways in which natural biology does life. So why do we bother making artificial cells on top of that, right? If we have so many different organisms to study. Well, the reason we're doing it is because all of biology, and I can safely say that as a trained chemist, all of biology is not a real science because you're using a sample size that equals 1, because we only have one life form to study. All of biology so far only studied a single life form because that's all we've got. There is only one life form on Earth. And despite this amazing kind of a diversity of form and function, there is really one type of biochemistry on Earth. And this is the rationale for the existence of our field, for why we're trying to build life from scratch. Because we want to build different forms of life. We want to find out how can life be done in a way that's not the life as we know it. So starting with what we do know, natural living cells are incredibly complex. There is 100 million or more proteins, different proteins in every cell. There is, depending on an organism, up to even 100,000 different genes. There are millions of small molecules that we don't exactly know what they are and what they do. So long story short, we've been studying biology since the beginning of science, and we still don't exactly know what goes into a cell. And so I'm going to use a extremely overused, but for a good reason overused, quote from a famous physicist, Richard Feynman. What I cannot create, I do not understand. And this is a perfect summary of why we're building synthetic cells. We do not understand life, and we will not understand life until we are able to build a living cell, a living organism from scratch. And this is the rationale for our field. We want to build a synthetic cell, an artificial living cell, and we're doing it so that we can better understand our own natural life. We're also doing it because it's inherently cool. We want to see that transition from living to non-living. And so to kind of fit with the theme of this series, Wilhelm mentioned robots. I want to say synthetic cells are essentially molecular robots. That's how you can think about them. They are biological components that behave in a very well-prescribed, well-defined way. So we do think about them as biological machines. And to me, who grew up prototyping and making my own little machines, synthetic cell engineering is kind of like biological breadboarding. We assemble synthetic cells from known components, and we see how those parts come together. How do we build them? How do they work together? How you can make a living cell from elements of other living cells or from unnatural elements. So now to dig a little deeper into the science for the next few minutes, I would like to show you on a molecular level what is a synthetic cell and what I consider a synthetic cell. So cells are named cells, as you know very well, after the compartment. The guy that first invented the microscope looked under the microscope and found those little square boxes, and he said they look like those cells that the monks live in. So let's call them cells. So the hallmark of all known biological life on Earth is compartmentalization, which means you separate entities. And so we do that in the most natural way. We build lipid vesicles. So a vesicle is a soap bubble. It's a liposome. Liposome is another word for vesicle. Don't ask me what the difference is because there's none as far as I'm concerned. The vesicle or a liposome is an entity made of lipids that self-assembles in a water solution around another part of a water solution. So it's basically a way to keep molecules separated. And all of your cells have those lipid membranes. So what we use to make our lipid membranes are those so-called phospholipids. So they're molecules that have a polar head group and a non-polar tail. And those molecules are self-assembling in water at the right pH to create those so-called bilayer membranes. They're called bilayer membranes because there are two layers of phospholipids in each of those membranes. We also love cholesterol. I'm sure you're familiar with cholesterol being bad. Every doctor will tell you how cholesterol is this worst thing you can do to yourself. No, it's not. Cholesterol is bad when we accumulate it in our veins, but it's really good when we put it in a membrane, and that's why it exists. It makes membranes much stronger and more fluid. So our synthetic cell membranes have cholesterol in addition to those phospholipids in their membranes to make the membranes very sturdy, very impermeable, but also very flexible. So now that we have a sturdy, impermeable membrane, we do want that synthetic cell to communicate with the external environment. So we add channels in that membrane. And those channels serve to bring nutrients from the environment, to read signals from the environment. And they also serve to remove waste products, as every metabolism, synthetic cells produce waste products. And the most common channel that we use in most of our work is this channel called alpha chemolysis. It's a very simple, very elegant pore protein. It's a peptide made from several simpler subunits, and it self-assembles in the membrane into this non-specific pore. And if the name alpha chemolysin sounds familiar to you, that's because this is also a technology that's gaining a lot of ground right now in sequencing. People use those nanopore alpha chemolysin derivatives and other similar proteins to make devices that sequence DNA and devices that, as of recently, can also sequence proteins. So this technology, even though we're using it to just make holes in a membrane, this also has kind of a practical applications. And I'll get back to that practical application area in a moment. So we have a membrane, we have pore in that membrane. But the whole point of a membrane is to separate the inside from the outside. And this can be done in a different way as well. So we, for most of our experiments, use lipids and make lipid membranes, but you can also not use lipids. You can separate your reactions or your synthetic cells using this so-called droplet microfluidics. So you can build those devices that create water-oil emulsions or water-oil-water emulsions that separate reactions from each other in those little droplets that look like liposomes but are not liposomes. This is a video of one of those examples of the microfluidic device made by my postdoc Theodor Christoforidis. And what we do here is we encapsulate a simple green fluorescent protein to visualize what we're encapsulating. And these guys look like cells, but they're just droplets. And this is another thing that you guys in the Netherlands are really strong at. Some of the best microfluidic engineering in the world in our area are people based in the Netherlands. So you have this amazing technology going. I was trained as a liposome biophysicist, so even though I do have microfluidics running in my lab, I much more prefer liposomes. So I will focus on talking about liposomes because they look more cell-like. But whether you encapsulate your synthetic cell in a lipid membrane or a microfluidic device, what's really interesting is what's inside a synthetic cell. So we encapsulate a central dogma. Right now, we're not fancy. We follow the DNA to RNA to protein central dogma. So our synthetic cells have DNA genomes. Sometimes they're linear DNA genomes, sometimes they're plasmids, but they're relatively simple genomes. Just to show you the scale of the engineering that we're doing, this is about the size of the human genome. This is approximate size of an E. coli genome. This is the first living synthetic cell that I'll talk about in a moment. That's the Craig-Venter mycoplasma-based synthetic cell. Now, ready for it? We are here. We are making those teeny tiny genomes. Notice that the y-axis is a log scale. We are happy if we can encapsulate 50 to 70 different genes, and that's the state of the art. So that shows you how, on one hand, early this field is. We don't have complex genomes yet, but we're very rapidly building up in the complexity. So in addition to the genome inside the synthetic cell, there is the whole central dogma translation machinery, this so-called in vitro translation system. So this is essentially guts of living cells, bacteria in our case, most of the time. We encapsulate all of the elements of the translation system. So ribosomes, we purify ribosomes and encapsulate them in synthetic cells. We purify the tRNA, all the other types of RNA, all the small molecules that you need, the RNA polymerase that's needed for transcription. We purify all of that and encapsulate in our synthetic cells. Those in vitro translation systems can come from a lot of different organisms. In my lab, we're lazy and cheap, so most of the time we use bacterial systems, but it's possible to use in vitro translation systems that come from a lot of different organisms. The more complex they are, the more complex are the proteins that you can make, but also the lower yields and the higher cost of experiments. So to build this kind of a complete synthetic cell, you have to put together all of those elements. And the beauty of this field is that synthetic cell is sort of like a jigsaw puzzle. All your elements do not have to come from the same source. We can combine things like ribosomes from one source, DNA based on another source, tRNA from yet another organism, and we can also use unnatural building blocks. So we can use molecules that do not exist in nature, and we do that. So just to show you how that thing looks, this is a microscopy image of a single synthetic cell taken in 3 different fluorescent microscopy channels. The green one shows you protein expression. The blue is a DNA stain, DNA dye, and the red is a lipid membrane. So they basically, they don't look like much. They look like those soap bubbles. They look like very simple cells under the microscope. And they're not alive. I really want to emphasize that what we are doing right now is not making a living cell. And I'll explain in a second why. So what we're doing is simple and is not yet living. And I know someone's already brewing a question of what is life and definition of life. And I'm happy to tell you we have none. And I'm happy to talk more about that later. But just for now, I want to tell you, we don't have a living synthetic cell, but a living synthetic cell exists. This is the JCVI mycoplasma-based synthetic cell. This is a cell that was made with a synthetic genome, but it is not made from scratch. The difference is that JCVI synthetic cell, it was the first living organism with a completely synthetic genome. It was made on a chassis of another living cell. It was made on a chassis of a mycoplasma bacteria. So it was never dead. To make the JCVI syn cell, you transplant a synthetic genome into another living cell. So that JCVI synthetic cell was never made from scratch from non-living components. So there is this unbroken line of life all the way back to the last universal common ancestor of all life on Earth. And so the JCVI synthetic cell has a synthetic genome, but they still don't know exactly what every gene in that synthetic cell do. They know the sequence of the genes, but they don't know the functions of all the genes. And that is different in what we're doing. We know exactly what we're putting into our non-living liposome-based synthetic cells. They're not yet living. We're working on that. We're building complexity, but we know exactly what goes in there. We have this complete component list. And that creates this kind of a friendly divide. I don't want to even say competition, just kind of a different approaches to engineering synthetic life. Some people take this top-down approach where they take a living cell and make it simpler and simpler all the way down to the point when we can actually understand it. And then people like me, chemists, take molecules, non-living molecules, we put it together and hope to build up a living cell. And the moment when we open champagne is when those approaches meet. So when the living and non-living meet and we actually build a completely understandable synthetic cell. Okay, so this is obviously a fascinating science, but I'm biased. Why would you care? Why would people like you care about it? So, we're doing those experiments in the lab. How does this actually translate into what can affect your life? What can affect people in the Netherlands, people all over the world? What are the practical applications of this? And when are we going to get there? That's a valid question to ask. I'm talking about it. When are we going to get there? So I want to show you a really funny quote. This is a quote from an article in the New York Times that was published on October 9th, 1903. And what they said is, it might be assumed that the flying machine might be evolved in from 1 million to 10 million years. Okay, so October 1903, someone in a very respectable journal predicted that it will be about 1 million years until humans can build a flying machine. And then they basically say, don't bother. They say to the ordinary man, it would seem as if the effort might be employed more profitably. So basically, don't waste your time trying to build an airplane. There are better things we can do. Okay, October 1903. 2 months later, the Wright brothers flew a first powered flight, the Wright Flyer in Kitty Hawk in North Carolina. So 2 months instead of 1 million years. And the ordinary man started profiting immediately. The moment we built airplanes, that revolutionized the world, that changed the way we travel, changed the way we do pretty much everything. So crazy scientific efforts are worth it. Crazy scientific efforts just look crazy. If you actually apply yourself, if you're crazy enough to follow it, you will do it. And to me, synthetic cells are the same. Right now, it might seem to you that building synthetic cells is going to take forever, and it's completely useless. But I can already show you some very practical applications of our field that are already coming to fruition, even before we actually build a completely engineerable synthetic cell. So some of those applications are, synthetic cells help us understand natural biology. By building a synthetic cell version of a specific biological pathway, we can understand how a natural cell works, and we can understand that much better. We also built tools to engineer synthetic cells, and those tools then go and help in different areas of science. The alpha chemolysin that I showed you 2 slides earlier, that was a protein used in the studies of the origin of life and building synthetic cells, and it spawned a complete new class of DNA synthesis technologies. If you are working in a molecular biology lab, you probably heard of the technique called Gibson Ligation. That technique was invented by Dan Gibson when he was working at the Craig Venter Institute, engineering synthetic living cells. So techniques developed in our field work and help to propel other fields forward. And then there are some practical specific applications. You can use synthetic cells to engineer new materials. We're using them to do biocomputing. We're using them to study the origin of life. We're using them for space exploration, for metabolic engineering, so for making molecules with biology. We're also using them for diagnostics and therapy. It's already been demonstrated that a synthetic cell can shrink a tumor in a live mouse. So it's possible to use those very simple, primitive cell-like structures for very practical applications. And I want to show you a couple more of those practical applications and things that can actually affect everyone, can affect basically every aspect of your life. So one of those things is the fact that right now we're depending on oil. And this is not a climate change talk. Let's forget climate change. Let's just talk about oil as a substrate. So when you think about what do you need oil for, you think about the usual suspects. Drive cars, make fuel, keep the lights on. We have energy from oil. We also heat our houses with oil. We also extract chemicals from oil. And so petrochemicals are the source of plastics. When you think plastics here, there's the usual suspect of plastics in your life. You think a little harder, there are a few more plastics you can name all around you. But there's also, you're obviously, as Dutch people, you're required to think about a bicycle. Every bicycle is made with parts that are derived from oil, that are derived from petrochemicals. Everything else in your life, electronic devices, shoes, clothes, everything you wear, everything you touch is made with petrochemicals. Medicine you take is made using petrochemicals. Also, we use petrochemicals to fertilize crops. We could not have modern agriculture without petrochemical derived fertilizers. We could not have commercial meat production without crop production. So that's affected. And also, me being Polish and having studied in Italy, I care about brewing, I care about fermentation a lot. That would also not be possible without materials derived indirectly from petrochemicals. So when you think about oil prices, you think about physically running out of oil, you think about energy. And right now we have amazing technologies for renewable energy sources. Those technologies already exist. We have to have them widespread, we have to have them widely adapted, but they exist. What doesn't exist is technologies to replace petrochemicals. And when we run out of oil, this is what's going to be the real economical crisis, because we do not right now have a way to replace all the chemicals that came from oil. And the only way we can think about replacing those chemicals is make them with a renewable source. So we already have a renewable biofactory, it's called a cell. We are a renewable biofactory. The problem with petrochemicals, though, is they're toxic. So no sane cell will make petrochemicals for you because they're toxic to natural cells. This is where engineering synthetic cells and synthetic biology comes really handy. We can engineer synthetic cells much safer than natural cells because they don't uncontrollably replicate. We can engineer synthetic cells faster, and that also makes engineering synthetic cells cheaper. So we can do all those things to hopefully produce biomanufacturing strains that will actually make those chemicals that we need to make us independent of petrochemicals one day. There's also a lot of other areas of science that benefit from advantage of having simple reproducible cells. If you're a researcher in any area and want to study biology, you know that natural cells are very noisy because every natural cell is different. If you study synthetic cells, the noise in your experiment is much less. If you ever run any metabolic engineering experiment or any fluorescent experiment, you know that signal to noise from natural cells can a lot of the time be very, very high noise for very low signal. So when you're trying to investigate some change in your experiment, whether it's biomedical experiment or biomanufacturing experiment, it's much easier to analyze that on a synthetic cell chassis because they're simpler. So that simplicity also translates into some kind of a far out there applications. So look at synthetic cell. You can digitize the information about making a synthetic cell. If you can digitize information, that means I can make a synthetic cell and send you a recipe for making that synthetic cells without actually physically sending you a molecule. And then you can make that cell. And so we can exchange information about making biological samples without the need to actually send biological samples. And this is obviously nice if I want to collaborate with Wilhelm. I can tell him exactly how to make my synthetic cell. But you can also say I can stick them in a FedEx envelope and send it to you overnight, right? What if Wilhelm was somewhere where I cannot FedEx something to him? For example, what if he became an astronaut? Or what if one of you became an astronaut? And I want to help you do an experiment, and then you need to reproduce that experiment. For example, because your crew got sick and you need to make a small molecule drug on a short notice. I, in my very well-equipped lab on Earth, can help you engineer a strain to manufacture it, and then send you information about making that particular compound. So that's possible with engineerable synthetic cells. That's not possible with living cells that you have to evolve. So people already took simple steps to show that this is viable. 3 years ago, we launched a very simple synthetic cell experiment into space. I'm showing that video because I had to get up at 4 a.m. to watch the launch, and also my own sample went to space. That was like super cool. I was really excited about it. We launched it. It didn't crash. It was a sample return mission. So we got to get our samples back. And we also showed that, yes, we can use those selfie systems to express proteins. So that opens up a lot of applications, not just in space exploration, but it also enables us to look for life elsewhere. If we build synthetic cells that resemble life, how under conditions that life could evolve somewhere else in the solar system, then we can move on and look for that life elsewhere. We will have a sample return mission from Mars. We'll have sample returns from and probably from Enceladus, probably from Europa within the next few decades. We'll find organics there. Do we know if it's life or not? We will if we can build artificial life and kind of recreate those artificial pathways to evolution. And then another very practical application of synthetic cell engineering is right now, if you get sick, you can take a biological drug, but it's made with those large natural cell bioreactors. So it's not possible to make a very customized drug for every patient. And you are different than anyone else. If you get sick, you should be able to receive a medicine that's personalized for you, not another patient with a disease that's similar to you. And that is becoming possible if we can actually have those small scale personal bioreactors. This is not possible if you have to build a giant natural cell bioreactor, but it would be possible to have truly personalized medicine if we can make those very small engineered cell bioreactors that can produce small amounts of drug for every patient. And these are some, not all possible applications of synthetic cells, but I hope I managed to at least give you a little bit of an overview of why this field is awesome, why we have so many different reasons to get into it, to get interested. And another reason why this field is awesome is because we are all nice people. Wilhelm already mentioned that. We're building this international research community. Europe has your own 2 different national synthetic cell organizations. South America has a synthetic cell engineering organization, but we're really hoping to bring the entire international community together. We don't have to compete. It would be much more practical, much better if we can actually work together instead of competing. And so this is the goal of our Build a Cell community, our Build a Cell organization. we want to bring people together. We want people to collaborate. We do it by sharing protocols, by having in-person meetings, by having online meetings. The last 2 years, we just had our first in-person meeting after the pandemic break. We're hoping to work together. You probably are aware that in 2 weeks, you're going to have in the Netherlands, in The Hague, this giant international synthetic cell conference. People are coming together, people are sharing unpublished results, people are sharing ideas. And this is what I really love about this community, that I believe that the moment we build a synthetic cell, it will not be a synthetic cell that's built in my lab or in Wilhelm's lab or in any other single lab. But when we build a synthetic cell, it will actually belong to the international community. Everyone who is helping with this community will have some part in the authorship of that first artificial cell. And in addition to having that final goal of having built life from scratch, we'll also be able to say that we did it as an international community. And that's what's really fascinating to me, is that it's not only fascinating science, but it's also a way to work together with amazing people. And I will wrap up the talking part of this meeting by thanking everyone who works with me. I'm incredibly lucky to have this amazing lab of people that want to work with me, that can put up with me and can put up with those weird research questions that we're asking. And most of all, thank you for coming. Thank you for inviting me. Thank you for talking. And I'm looking forward to the questions. And if you want any more information, we have nice websites. So please look us up. Thank you.
Wow, Kate, that was a fantastic talk, a visionary talk, really very impressive. Not only because of the vision, but also because of the fantastic graphics. I really enjoyed that.
Thank you so much.
I enjoyed listening to it and I enjoyed watching it. So that was a great presentation. One question you didn't answer, because you showed us why you should build a synthetic cell. And I think you have given us many convincing and visionary and exciting opportunities that you could think of when you have a functional synthetic cell. But is it going to take 1 million or 10 million years to build this synthetic cell? Or do you have a better number for us?
So let me answer this question this way. In the US, when you're hired as a professor, you're hired in this so-called assistant professor position. And you don't get to stay unless you do something really good. That's called a tenure process. And so my tenure clock is ticking. I'm up for tenure next year. So that's the deadline. We have to have a synthetic cell. Seriously, though, I really think we're a couple years away. Thanks to mostly you guys. One of the last big remaining things in synthetic cell engineering is replication. And some people in the Netherlands are really pushing the envelope of what mostly actin-based filament replication can happen. You guys made those contraction rings already. There, I think we'll get replication. We already have gene expression. We have other elements. So I think we'll be able to say that we sort of made a living synthetic cell, you know, within anywhere between 2 to 5 years. That's my guess. And if you ask me next year, I will again tell you 2 to 5 years. And I'll keep saying that until we're there.
Yes. If you ask for a 10-year program, then as we did in the basic program, then we of course say it takes 10 years. But there you go. No, I appreciate that. There's lots of people all around the world making different modules. And especially replication is a tough one. But as you say, it's a very essential one. I have many more questions, but I'll give people in the audience a chance as well to ask questions, and I'm sure Amy will take care of that. In the meantime, I have one question via YouTube, which is a really difficult one, Kate, because it means you have to have been watching the previous episodes, because Marcel Blattner asked, what is your take on Xenobots? And I don't know if you watched the Xenobots episode, but Josh Bongard from the University of Vermont talked about these clusters of cells that he could actually simulate first in a computer program,
Yes.
And then stick together or snip bits and pieces off clusters of cells, and then they were going to contract and push and expand and actually have similar kinds of movements. So I guess the question is, what is your take on this?
I think this is an essential and necessary step towards making a actual synthetic cell. So to me, because there's no good definition of life, I think von Neumann's universal constructor is a very good approximation of what life is and what a self-replicating machine is. And I think once we make synthetic cells, they'll essentially will be those little xenobots. People that are building those xenobots right now, they're not using all goo. And that's why this is not a cell for me, because to me, a cell needs to make all of the components of itself. But I think they're definitely on the right track. And the things they're inventing, especially in the realms of control theory and in terms of assembly, will definitely contribute towards making a completely autonomous artificial cell.
Cool. Yeah, I can see your point there. I know that we have a question in the audience, so I'm switching to Amy.
Hello, can you hear me? Yes, okay.
Yes.
And my question is about the use case of using synthetic cells to create chemical compounds. Because as far as I know, it's already possible to engineer a genome to make some chemicals and then put it inside, let's say, a normal cell to produce something. So it's not clear to me what would be the advantage of creating a synthetic cell to do that instead of, let's call it, the more traditional approach of modifying some DNA or creating a new DNA and then injecting it in some cell that we already have.
Thank you very much for that question. Yes, that question emphasizes one of the biggest advantages of synthetic cells, actually. They start out dead and we design them from scratch. So you're right that we can engineer natural cells to make a lot of chemicals right now. But if a chemical that we need is toxic to natural life cells, they will not make it. You can engineer any enzymatic pathway you want, and you can keep engineering it till you have to retire. The cell will not make the compound from your pathway if it kills it. Or if it makes it, then it will immediately die and stop making it. And that's the biggest problem in the biofuels and among the people that try to make all those toxic compounds that our entire life is based on, the petrochemicals. Natural cells will not make something that will kill them. You see that very well in the process of brewing. You cannot brew alcohol to a higher concentration than the yeast can live in. If you want higher concentration, you have to distill your alcohol. And the same with chemicals. You cannot make chemicals that are toxic to the organisms that make them. And because it so happens that we organized our entire society around chemicals that are awfully toxic to life, it's really hard to make those chemicals using natural cells, because no self-respecting natural cell will go and make those chemicals for you because they'll just die. And so engineering synthetic cells, we can engineer the entire metabolism around the chemical that we need to make. So for example, if we want to engineer a chemical that is toxic to the ribosome, we first have to engineer a ribosome to remove the vulnerability that makes the chemical toxic to that ribosome. And then we put it in a synthetic cell, because then it's an engineered ribosome, so any cell that carries it will be synthetic. And then we can make the toxic chemical that we need. So for most applications where the product is not toxic, natural life cells work perfectly fine. But now we are arriving at the needs where the chemical is toxic. And for that, we need to actually engineer new cells.
Kate, if I may, I can see your point of the factory of the future, exactly for this point where you say making chemicals that the cells would not be able to do because it's toxic, or indeed they might do it in such low yields that it becomes no longer economically viable. What I did not quite understand was this idea of teleportation, because I can send you the code, but then what? The code still has to be translated somehow into molecules. How do you do that? How do you envisage that?
So the basic requirement for that to work is us working together to establish a common chassis. And this is kind of what the Build-A-Cell is trying to do too, is we basically have to agree on what molecules we're going to use to build our synthetic cells. So what ribosomes, what lipids, what small molecules, and you should have a DNA synthesizer. So once all of those things are in place, then I can tell you synthesize that gene, put it together with that concentration of ribosomes, that concentration of amino acids, and so on. So it requires this common language.
So you would assemble the new synthetic cell on the spot, essentially.
Yes,
it's not only the DNA, but also all the other ingredients that you need to put together. Okay,
we need to first agree on this common language, and we need to have the ingredients, which is why, you know, it takes preparation. I'm calling it teleportation because I'm a sci-fi nerd and I like the word, but it's more like, you know, making remote assemblers, sending information about how to assemble something remotely.
Yeah, that's an interesting thought for sure. There's another question from the audience, Amy.
So first of all, thank you very, very much for this inspiring presentation. It really touched something inside and my nerves. And my question for you is about the form of the cells. So you are trying to make a synthetic cell, and you are based it more or less in the characteristics of our cells, which will be in form spherical or a bit elongated. But can you think about other shapes or forms that a cell could have and how it could enhance or decrease the efficiency or the functionality of the cell?
Absolutely, and people are already doing it. My synthetic cells are mostly spherical because we just make simple liposomes, but that's actually another area where the Dutch people are leading the way. There is a lot of work on internal synthetic cytoskeleton done in the labs in the Netherlands and in some other places. And so people are trying to shape synthetic cells into different shapes using the same tools that biology uses to shape cells. So using proteins to build a cytoskeleton. And then you can have a rod-shaped cells, or, you know, one day maybe we'll be able to make cells that look like budding yeast, for example. So yes, there's definitely efforts towards that.
I think there's another question in the audience.
Yes, thanks. First, thanks a lot for your great talk, Kate. And I have a question: is that how do you define the living? So, to what state do you think the synthetic cell is a living cell? So now we can do that transcription and translation. And what's the next step?
So first of all, I have to start by saying I do not have a complete definition of life. I cannot tell you what is the definition of life because no existing definition of life is complete. NASA definition of life, which is one of the more commonly ones used in the field, states that it has to be a self-replicating system that's capable of Darwinian evolution. This is a good working definition until you start thinking about it. According to that definition, you are not alive because you, by yourself, cannot self-replicate. And I'm not alive because I cannot have a baby on my own. I cannot self-replicate. So according to that definition, nothing other than single cells is alive. So that just shows you that there will be some shortcoming in any definition of life. So to me, the kind of a hallmark of when we're done is a quote from a United States Supreme Court justice who said, "I will know it when I see it." That was obviously not used in a context of biology. That was used in context of pornography because he was asked to define what's pornography. And he said, "I don't know, but I'll know it when I see it." And that's actually what we're doing, at least what I'm doing. I don't know at which point my synthetic cell will become alive. But when it becomes alive, I'll know it. I hope I'll know it. To me, there are some hallmarks like self-replication, ability to evolve, ability to sustain a cell cycle, ability to sustain homeostasis. And once all those elements come together, we'll make a cell that hopefully will start doing most of the functions of life. And then most people will agree it's alive. I don't think this will be a light bulb moment. It will not be that like one day I'll come to the lab and say, oh, I made life. No, it will be a very prolonged process over probably several years when people will be publishing more and more complex synthetic cells. And at one point, we'll all look at each other and say, hey, this actually is alive. And I think that's going to be very anticlimactic, but also very satisfying. And I realized I did not actually answer your question, but there is no good answer.
Well, I must say, we have been guilty a little bit of running 2 seasons of late night conferences on origins of life and synthetic life without addressing the question what life actually is. But I will sort of already warn you, next month, we will actually have a speaker who knows more about this. There are more questions in the audience, though.
Yeah, so thank you very much for the very nice talk. When I was looking at the picture with all the labs spread around the world, I was wondering, will this synthetic cell, which has such an open source origin, once we will be able to use it for biotechnological application, will it somehow change the way patents are made? Like, will it change the way we, the way we address the merit of this discoveries and applications? What do you like? How do you envision that?
The optimist in me says I would love if that happened, but the realist in me says it probably nothing will change. Every field gets to the point when we need money beyond the research funding, when we actually want to build applications. And at that point, we need private money, we need investors. And to attract investors, we have to have something that we can give them. So I honestly think that technology will become open source, will stay open source, but the particular applications will be patented. I think it's likely that we might follow the CRISPR route, where the research applications are open, the specific biomedical applications are locked by patents.
Thank you, Kate. There are more questions, I think. Amy?
Coming. Go.
Notice that they're not throwing the microphone, just like you asked.
Well, we are doing that, but apparently not. We have Amy running around today.
So thank you. You showed a video of an experiment you shot in space. I was wondering what exactly did it investigate and what would its use for space exploration be?
Thank you. So what we investigated was whether a lyophilized cell-free protein expression system can be used under spaceflight conditions. And so that involves all of those practicalities of launching mass into space. So it has to sit on a tarmac in Virginia in the summer, which is very hot for a few days before launch. It has to survive the launch, which is essentially a very expensive paint shaker. And then it has to survive the rehydration. And we sent 2 experiments. One was to investigate folding of a functional RNA made in vitro. So we made an in vitro aptamer, RNA aptamer called broccoli. And the other experiment was in vitro translation system. So we investigated whether we can make a protein. And we made a very small protein that can be observed using green fluorescent channel. So this was proof of principle that shows that basically those systems can survive spaceflight conditions. We haven't made anything interesting yet other than a glowy green protein, but this is our next step. We want to actually send liposomes, whole synthetic cell liposomes, and see if those experiments can be done under human spaceflight conditions.
Thank you, Kate. Let's hand over to the next person who is asking a question.
Dzień dobry.
Dzień dobry.
my question is that you already talked about biological teleportation, and I wanted to go a bit beyond that because I know there are capsules on, for example, abandoned satellites that have some information about Earth and people in general, kind of coded like in a mathematical way so that you can uncode them even if you don't know what's that, just based on apparently basic physics and basic mathematics. And my question is, if there's actually a recipe for the synthetic cell present one day, do you think it would be better for it to stay on Earth, or would you send it with the capsule either way?
I would send it to whoever wants to receive it. You know, if I would broadcast it on every channel, if we still had the Arecibo telescope, I would send it out. Let aliens make a synthetic cell. Why not? Seriously though, I think one of the main guiding principles of our field is openness. We share protocols, we share information. So at the time when we're ready to actually have a recipe for a synthetic cell, I want it to be shared. I want everyone to use it. Obviously use it responsibly, and this is another kind of a field of research that we're working on, is safety, security, and sharing of information. But yes, I think we should be sharing, we should be broadcasting it.
I think Amy has a question, or Anna has a question.
Hi, Kate. Thanks a lot. Whoa, my bad. I have a quick question. I'm curious about. I'm going to come to the camera so I can see you. I'm curious if when you build the synthetic cell, do you imagine it's going to become so complicated that you might actually need lots of statistics and stuff to figure out what's actually happening in this more complex interconnected system? And do you think that could be then almost like we're looking at a living system? Do you think that's when we reach a point that we find life? Or yeah,
yes, I really hope so.
Okay.
I really hope that it will become complex enough that it will not be defined by simple biochemical reaction properties. We call this emergent properties, when the sum of the parts is more than a simple sum of the parts, when there are some new properties coming out of it. And then we're going to need the math people to help us analyze it. And I'm really looking forward to that.
Awesome. Yeah, thanks a lot. And I think Anna has the next question, but you might have to go up to the technical.
Yeah,
booth.
I will read it for you. That's okay.
Thanks.
Anna Knox has a question, and she says, which scientific advance, other than your own research, has had the most impact on your work?
That's a very difficult question, because everything I do builds on others' work. You know, this saying that science is standing on the shoulders of giants, that's very true. Everything I do is a little addition to someone else's work. So there was foundational work in the technology development. So for example, development of those cell-free systems. People like Vincent Noiro, who made those cell-free protein expression systems actually accessible. Those people paved the way for our field. People like Wilhelm and other people in the Netherlands who built the technologies to actually assemble those systems to help us understand the behavior of the compartments. They helped us even imagine what's possible to do. People that work on protein engineering help us figure out what are the limits of types of proteins that we can make in those synthetic systems. People that work on DNA replication in synthetic cells provided mechanisms to replicate the genomes of our synthetic cells. So everything is basically building. Everything we're doing feels like incremental progress, and it is incremental progress. It's not a single person that solves all the problems. It's more like a hive mind. So everything I do builds on work of other people. So I can only say there is, about 50 years of work before I even started my lab in this field. And I'm just adding those tiny little pebbles to the pile.
Thank you. Will Robinson has a question, and he says, every scientist has individuals who are important to their career. With regard to those who have helped you over the course of your career, how did you meet and develop the professional relationships with them?
I was very lucky. The people I met along the way are people that were around when our field was starting, and those basically founding fathers and mothers of our field. And I was very lucky to meet them during my undergrad. I met both of my grad school advisors when I was still an undergrad, and I learned a ton from them, not just about science, but also about connecting with people. Through the work in the Build-A-Cell community, I'm meeting all those people whose papers I've been reading, but I never met them in person. Now I'm meeting them and they're all nice. There must be some filter on our field, because a lot of people in our field who I'm meeting, there's those giant people that I read their papers, they're famous people. I meet them and they're actually nice and they're willing to talk to me. So that, a lot of it is luck, but a lot of it is also being bold and brave and cold emailing people. That's how I met a lot of people when I was just getting started. I was just sending emails saying, hey, I think your work is fascinating. I have a question about your paper, or I have a question about research directions. And people took time to respond to me. And that's how some of my best long-term research relationships started, is just me cold emailing someone. And this is one of the things I like about our field, is most people are really nice and friendly.
Thank you, Kate. I think the questions in the audience have dried up, but I have a very important question to you. It's a bit of a technical question. It's a kind of Pepsi versus Coke question, and that is built on the very sort of structure of that synthetic cell. Are you going to use cell lysate, or are you going to use the pure system?
Cell lysate all the way. I'm a firm believer.
You're my friend.
Yeah. The reason for it is because I'm lazy. I think to really make something that's robust and widely used, we have to rely on technologies that are not prohibited in any way. They're not too expensive. They're not too time consuming. Those experiments are hard enough as is. We don't have to make our life any harder. And also, cell lysate comes with some built-in complexity already. And we're taking advantage of that complexity, because pure system gives much lower yields, and it's much harder to engineer specific elements of complexity into it. So yeah, I'm definitely a lysate person.
Okay, I really hope that Christophe Danelon in Delft University has not been listening tonight, because otherwise your trip to the Netherlands in 2 weeks' time is not going to be an easy one. No, but we should definitely have a discussion about that, because to be fair, of course, if you're making a well-defined synthetic cell, then having a well-defined so-called pure system instead of this very messy and complex lysate is, well, logically speaking, actually quite a good step. I think I agree with you. Practically, there's disadvantages. Yeah. But the pure versus lysate discussion we can have over a beer in The Hague in a couple of weeks. I think for now we are going to close this evening. Kate, I would like to thank you once again for a beautiful and inspiring presentation. I've really enjoyed this one. I hope that everyone who has been watching online or in the audience has enjoyed it as much as I did. I would like to thank the team once again for making it work today, when we had once again an unusual situation with me not being live, but the audience live, and Kate zooming in from Minnesota, and I think everything went extremely smoothly. I also already hinted at our next speaker in 1 month's time on our usual Thursday evening, which will be a philosopher, and she will be discussing what is life with us at the very end of 2 amazing seasons on artificial life and origins of life. That's it. That's all for tonight. I thank you all once again, and everyone who is in the audience, go and grab a beer at the yard. So with that, I leave you all for tonight. Goodbye.